So yeah, so in order to explore the sequence landscape, we propose that we can use deep learning in order to do this. And this takes advantage of many of the advances that deep learning has provided us with, especially in the protein space. So specifically what I'm talking about is our deep learning models that are able to basically go between sequence and structure. And so here I'm just showing you a diagram. We can go from the forward direction. If we go from a sequence to a structure, this is what AlphaFold does. In one direction, going from sequence to structure are things like AlphaFold. But what's also really exciting is now that we can take a protein structure and we can go in the reverse direction. So these are models that you might have heard of, the most prominent being something called protein MPNN that came out of the Baker lab. And so what you can do now is just give these models a structure of a protein kinase that you might be interested in, and then it can propose, sequences that can fold into that kinase sequence. And so going back to this kind of figure, really the specific question that we're interested in is, can we use these types of inverse folding, so going from structure to sequence, in order to access, new parts of sequence space, and then can we measure those, in order to be able to really understand what the sequence function landscape looks like for, a given protein kinase.